Hi, my name's Ramesh. I will be presenting for our group on the effect of varying light intensities and food sources on planaria. So planaria or flatworms are commonly found in bodies of fresh water, such as lakes, ponds, but they're also found in wet areas or environments such as muddy and rocky habitats. They're usually carnivorous invertebrates, and they feed on shrimp, snails, and other animalia tissues. The photoreceptors in their eyes does react to light stimuli, and it allows them to move by the means of negative phototaxis. So they move away from the light, and they have been found to prefer artemia over liver, and their rate of ingestion is increased in lower light conditions. So our experimental question was, how will the intensity of light that the planaria is subjected to affect the eating habits? And if we hypothesize that if we decrease the amount of light that the planarians are subjected to, then they will increase their rate of ingestion, and they will also prefer the artemia rather than the liver. And we predicted that in the presence of low light, the planarians will be able to locate the food source, and they will prefer the artemia and travel more efficiently when compared to brighter light conditions. So these are just some of the materials that we can look over. You can pause the video there as well. So we had a total of, I believe, 9 planaria. So 3 different trials subjecting 3 different planaria at a time to different light intensities with 2 different food sources, dead artemia and liver. So for high light and intensity, a microscope was used, turning the brightness to its full capacity. And for lower light conditions, the planaria were wrapped in a container, and they were given some time to adjust to the dark. So the control variable is the number of planaria and the type of food source, while the manipulated is the light source and the varying light conditions. And the responding variable we did was the time taken to get to the food source and which food source the planaria preferred. So this is the darker light conditions and the brighter light conditions. So some of the results. In the room light condition, as we see, there's a medium amount of time that it took to find the food source. The brighter light condition caused a total of 7 minutes, the most amount of time to find the food. And in low light, the one they preferred, a smaller amount of time, 1 minute and 36 seconds. So here with the lower light conditions and in the dark environment, all 3 planaria chose the artemia to eat. In mid light, 2 artemia and 1 liver, while in bright light, one chose artemia, 1 the liver, and 1 just roamed around and couldn't find a food source. So the planaria, they seem to strut under brighter light conditions, as was depicted when they moved much slower, and movement was observed through gliding, and the planaria did feed by sucking food through their pharynx. So the hypothesis was confirmed, and when planaria were in low light, they consumed more food faster, and specifically they were driven towards artemia rather than the liver. And brighter light caused the planaria to actually not go towards any of the food and left them confused. And they had the highest level, the highest number of minutes when trying to find the food. And they preferred darker environments due to the genes in their cells that react to light, also known as the photoreceptor cells. So when exposed to bright light, the photoreceptors are temporarily impaired, causing them to not see as well in bright light conditions and not finding their preferred food source. And other researchers have also discovered that the planarians need low light for getting nutrition and reproduction, and they've evolved in a way that they can access nutrition and survive without the need of light. So future improvements. To measure the regeneration, we can also measure the food and label the actual food, such as the shrimp and the liver. That way we can see how digestion occurs. And then we can use a light meter to measure the different intensities of light, just so it can be a bit more accurate. And we can also increase the sample size. We only had 9 planaria. This will definitely help reduce bias. So people are often using planaria as pets. We can use the study to ensure that we're keeping these cute planarias under optimal survival conditions and giving them artemia instead of liver, as they enjoy that more. And planaria are also grown in labs for different experimental conditions. So the study can be used to make sure that they're living in optimal conditions there as well. Thank you, and here's our literature cited.